Hi everyone, welcome to my channel Neurobiology, and today I come with another topic, that is the exocytosis. Okay, and this is the related topic, the function of plasma membrane. Okay, and in my previous video I have already explained about the endocytosis, and here I will try to explain about the exocytosis. Okay, and before going to this topic, I want to share that please share, subscribe my channel for getting the notifications. Obviously hit the bell icons. Okay. So let's continue. This is the exocytosis. So what is the exocytosis? Exocytosis actually here the transport vesicles that destined for plasma membrane undergo fusion with the plasma membrane and release the contents outside the cell. So endo means the internalize the cell and exo means that is the exit from the cell. Okay. So here what happened, that transport that vesicles fuse with the plasma membrane to release its contents. Okay, the vesicles that containing maybe proteins or hormones or enzyme etc. So it just fuse with the plasma membrane of the cell and that release from the cell. Okay, that the contents that released from the cell. This is called the exocytosis. By the process this is happening, this is called the exocytosis. So generally in this exocytosis there are different method or different process or pathway is seen. Okay, different pathways generally are seen, like that constitutive secretory pathway, second regulated secretory pathway. Okay, so constitutive secretory pathway that is occurring by the cells and regulated secretory pathway that is occurring by the specialized cells. Okay, constitutive means continuous. This process is occurring continuingly. Okay, but this regulated secretory pathway, this is not occurred continually. It is occurring sometimes, but with the help of or by some specialized cells. Okay, not all the cells are capable for doing this regulated secretory pathway. Okay, but generally constitutive secretory pathway that is possible, or all the cells generally they are capable for doing this, but this is not possible for all cells. Some specialized cells that are involved in this pathway. Clear? So example of proteins of constitutive pathway or constitutive secretion, that the collagen by fibroblast, examples collagen by fibroblast, antibodies by activated B lymphocytes. Okay, so these are the examples of constitutive secretions. Okay, so first one, collagen by the fibroblast, this is one example of continuous secretion, and another example is the antibodies released or antibodies activated, antibodies released by the activated B lymphocytes. Okay, because only the B lymphocytes, they activated B lymphocytes, they release or they tend to release antibodies. Okay, and the regulated secretory pathway here, these are found by the specialized cells. So here for secreting products rapidly on demand. That means if any content or any secretory substance is necessary for the cell or for the body, that at that time this pathway is occurring by some specialized cells, like that hormones and neurotransmitters. Okay, these are released by this pathway, regulated pathway. Where is that line? Regulated secretory pathway. Clear? So here the secretory pathway, the secretory vesicles are formed from the trans-Golgi network. This is very brief or rough structure of the Golgi complex, and this is the trans-Golgi network, and here the two pathways are seen, and first one is the constitutive secretory pathway and second one is the regulated secretory pathway. And in the secretory pathway, what happens? First, these vesicles are formed from the Golgi, trans-Golgi network, and they release their contents to the cell exterior by exocytosis. You can see that the vesicles are formed with some contents, and then these are fused with the plasma membrane, and then the contents are released from the cell. Okay, and here in response to specific signals, okay, this is very important. When the signal is very needy for any secretion, that then it is obviously under the regulated pathway. Okay, secretion by the regulated pathway. And when the signals are not necessary, that means the continuously secretion is occurring, then in that situation this is called the constitutive pathway. So what is the main difference between this constitutive pathway and the regulated secretory pathway? The main important difference is that here one signal, in one case the signal is very important if or until the signal is alert, the regulation or the secretory substances are not released. Okay, but in case of constitutive secretory pathway, there no signal is necessary. So here the secreted product can be either a small molecule. Okay, secreted product may be the small molecule such as histamine, example, or a protein, maybe protein, hormone or enzymes. Okay, and protein destined for the secretion, that is the secretory protein. So which proteins that are destined for the secretion, these are called the secretory proteins. And obviously these are packaged into appropriate secretory vesicles in the trans-Golgi network, and specific signals direct the secretory proteins into secretory vesicles. Why this signal is very important? Because these signals direct the protein substances or protein contents to enter into the secretory vesicles for releasing from the cell. Okay, so that's why signal has important here. Okay, there is signal is very important. Why? So if signal not direct the secretory proteins to enter into the vesicle, so the secretion is not possible. That's why signal has importance here. Okay, so these are the two processes I just briefly sketch here, and you can understand what I am saying in this process, this exocytosis process. So this is one function or another function you can say about of the plasma membrane and or the cell membrane. So in my next video I will try to explain another topic related to obviously cell biology. So continue with me and my channel. Thank you.